Welcome to Study Loop.
Here we are looking at RNA stability. So these are going to be the RNA molecules that have already been processed. So these have been spliced already, and we are going to add a 5' cap and a poly A tail as well. And there can be the additions of certain RNA binding proteins, which are going to be proteins that either increase or decrease the stability and activity of the RNA molecule. So you can imagine that you have your regular RNA molecule that has already been spliced. So this is going to have several different regions. You have several different genes associated with the RNA molecule, and you're going to have something called the untranslated region, which is not an intron because those are removed during splicing, but this is just a region that does not produce a protein. So it will be after that stop codon. So this has just been processed inside of the nucleus, and we are going to add different products of that 5' cap, as well as the poly A tail. And those are going to increase the stability of the RNA molecule because the 5' cap helps protect the beginning sequence and is going to tell the ribosomes where to start. And the poly A tail is to prevent the digestion of the important nucleotides by the enzyme RNase. And instead, RNase will digest these useless adenine groups added on to the end. In addition to these, we of course have those RNA binding proteins, which can consist of several different types, and these can have several different effects. So they can just bind typically in the untranslated areas or before the important genes of the RNA sequence. And those are going to either increase the stability, or in some cases, they can decrease it if they're going to promote the digestion by an RNase or say by a proteasome. So here, because these binding proteins are able to do both of these functions depending on what type they are and what effect they have, the best answer choice here is going to be D, that your RNA binding proteins can both increase or decrease the stability or lifetime of that RNA molecule.